Okay, we'll make a start. Look, good evening, everybody. You're all very, very welcome to this webinar. My name is Andy Welton, and I'm your host here this evening. This is the first of a series of webinars we're going to run over the next number of months. And yeah, delighted you're able to join us tonight. We're joined by a number of people, and I'd like, first of all, to introduce my colleagues. Most of you will know Owen Sweetenham. Owen, you may put up your hand there and give us a wave. Owen is one of our key specialist vegetable advisors based covering the eastern side of the country, working out of Ashtown in Dublin. And I'd also like to welcome a new colleague to our team, William DC. William, you may put up your hand and give everyone a wave. William has recently joined us, ladies and gentlemen, and he comes armed with, a PhD in the whole area of integrated pest management. He's a specialist veg advisor. He's going to be working out of the Teagasc office down in Ennis. And he's going to be focusing some of his time also on the kind of organic side of the house. So welcome to William, and great that you're with us. We're also joined by two other very important people tonight, ladies and gentlemen, our keynote speakers, and you'll have seen their names on the opening slide. We have Dr. Lauren Chappell from Warwick University, and we have Colin Noble from Veg Consultancy Services in Norfolk. Welcome to you both, and they're both experts on the topics that we're covering off here tonight. But maybe before we come to that, I just want to draw your attention to a couple of the rules that go along with these kind of meetings. For anyone familiar with them, you can hopefully see us all here tonight and hear us loud and clear, but we can't see you. But look, that doesn't stop you guys from engaging with us, and we'd like you to engage. And the way you can do that is by putting questions to our panelists and our speakers. And there's a little Q&A tab down, usually at the lower end of the screen there. If you open that box, you can enter your comment or your question there, and that'll come to ourselves here. And William and Owen will take those questions, and they'll screen them, and we'll field them to our speakers over the course of the evening. We're recording the webinar, and in case you want to look back on it at some stage in the future, or if you know someone that mightn't have been able to join us tonight, that recording will be up on our YouTube channel in the next week or 10 days, so you can view it there. We've also got some IASIS points or credits going with this event or this meeting. So for those of you that are interested in being awarded those credits, Owen will put a link so that you can fill in your details in the chat box. Again, you can see that icon down at the bottom of the screen. If you click that, you'll be able to put your details, and we'll make sure that you'll be rewarded with the IASIS points for attending the event. So the plan is to cover off, like I said, both of these important key diseases. The number one disease in carrots being cavity spot, and canker, which is number one on parsnips. And the intention is to hopefully get through that by way of presentation from our expert speakers over the next hour, 10 minutes, hour and a quarter. So we'll aim to try and finish maybe around the 8:15 mark. So without further ado, I'm going to stop sharing here, and I'm going to ask Dr. Lauren Chappell to perhaps share her screen. And while she's doing that, I'm going to just introduce Lauren and tell you a small bit about her. Lauren is currently a research fellow at Warwick Crop Centre at the School of Life Sciences in the University of Warwick. She graduated with her primary degree in biology from Cardiff University, and followed that by a master's in molecular plant biology at the Imperial College in London. and the Royal Botanic Gardens. She then obtained her PhD in plant pathology and plant breeding, focusing on parsnip canker at the University of Warwick. And after that, she worked for a period as a plant pathologist with Elsem seeds. She went back into academia, then working on Pseudomonas syringae, which is an important bacterial plant pathogen. That was at Oxford University, before returning to Warwick in recent years, where, like I say, she's a research fellow working on the DEFRA-funded Vegetable Genetic Improvement Network. And I think, Lauren, you'll explain a little bit more about that in your presentation. So with that, I'm going to hand over to Lauren. We'll quickly follow with Colin's presentation, and what we'll do after that, we'll take questions on the cavity spot element, and then for the second half of the evening, we'll focus a little bit on the parsnip canker problem. So thank you, Lauren.
Thank you very much for that lovely introduction, and thank you for the opportunity to speak this evening. Hopefully you can all see and hear me okay?
Yeah.
Excellent, thank you very much. So this evening, obviously, we'll be talking about cavity spot on carrot, kind of thinking about developing techniques for research and disease management. So I thought I'd just start off with a little bit of background about the project and the kind of approach that we're coming from. So I work on the Vegetable Genetic Improvement Network, or VEGIN project. It's a DEFRA-funded project, which is very much a kind of interactive network of researchers and industry leaders. The aim is that we kind of all work together to identify key novel genetics for improved traits, for kind of sustainable production in key vegetable varieties. We work with the 4 key UK vegetable crops, obviously carrot, onion, lettuce, and vegetable brassicas, and really is all about kind of using and identifying genetic diversity, key genes linked to key traits. So for us, that's mostly resistance to pest and disease, or kind of tolerance to climate. So why is genetic diversity so important? I'm sure you're all familiar with the kind of ancestral carrot. And whilst nobody wants to be eating that or growing that today, from our perspective, that's actually really important because that contains loads and loads of different genes. If you think of the breeding process a little bit like going through a funnel system, each time you make a selection for a specific trait, you focus on one specific gene and get rid of everything else. And as you kind of go through this funnel system, it means that the modern carrots are brilliantly uniform, high vigor, which is brilliant for you guys. But from a research perspective, we've lost a lot of that genetic diversity, which is why we need to kind of go back to the beginning and use those original genes to identify sources of resistance and tolerance. So here at Warwick, we have the UK Vegetable Gene Bank, which is a seed bank all about conserving and using vegetable crop diversity. Huge numbers of seeds and accessions, over 520 million seeds from over 108 different countries. I think over 14,000 different accessions. Of course, we're interested in carrot, and we have about 1,500 accessions of carrot. This is an awful lot of carrots and a little bit too much for us to be working with on a daily basis in terms of research. So within VEGIN, we've curated what we call the carrot diversity set. So this is a subset of all of those carrots held within the gene bank. We have 77 lines that kind of capture all of that genetic and morphological variation present in the collection. So it kind of captures all the different root shapes and colours, all sizes, captures the variation from across the globe, different foliage characteristics. You kind of name a trait and we have an example carrot in our collection. And that's made up of wild species, kind of land races and traditional varieties, as well as advanced and elite commercial varieties available. So we really have a huge range of genetics available for us to test and assess against. So cavity spot, I'm sure I don't need to tell you some of this, but we'll cover the basics. As you all know, cavity spot is one of the biggest problems for carrot growers in the UK. Estimated losses are currently at around £5 million per annum. It's principally caused by the Oomycete Pythium violi, but we know that there are others involved to a lesser extent. Things like Pythium sulcatum, even intermedium and sylvaticum. I'll go into those in a little bit more detail later on. We know that other pathogens can cause similar root lesions and invade the cavities, things like cylindrocarpin and mycocentrospora as kind of secondary infections. They kind of lead to quite different symptoms, so they're kind of secondary infections to the original cavities. Often be misleading because you in fact don't think it's cavity spot, but cavity spot was the original infection. So what are the problems? Well, we need to understand the interaction between components of the cavity spot complex. We know that there are different pythiums involved, but we don't really understand how they work together yet. We know that cavity spot is unpredictable. You know, different weathers can instigate different levels of disease. And we know that pythium itself is unpredictable. We don't really understand the timing of the infection yet. And of course, the issues around control, metalaxyl, the lack of new actives, and therefore the need to kind of identify varieties with good tolerance and resistance. You know, Nairobi is currently the most widely grown carrot, I guess the industry standard, if you like. And if we're honest, that's got low to intermediate resistance. So in this presentation, I'm going to talk about some of the kind of techniques that we've been looking at and developing. First off is identification of pythium isolates associated with cavity spot. If we're going to be breeding for resistance, we really need to understand what we're breeding for resistance against. And secondly is development of an artificial inoculation system to induce cavity spot. This enables us to test new crop protection products, fungicides, as well as biocontrols, and also identify new sources of plant resistance. So thinking about the pathogen side of things, Pythium violae really is the major cavity spot pathogen in the UK. So we collected cavity carrots infected with cavities from around the UK and plated them up and tried to identify what the causal agents are. And as you can see here, over 50% of the cavities are caused by Pythium violae, with intermedium, sulcatum and sylvaticum kind of coming in as a close second. What's interesting is when we carry out root inoculations, Pythium violae cause the biggest lesions by far, followed by sulcatum and intermedium. You can kind of see that progression of lesion sizes along the bottom there. So this really was the most important first step, starting to understand that complex of pythiums and which kind of pathogens we really need to target when we're looking for control and resistance. So next up, we're trying to develop kind of pathogenicity assays. Can we get something growing in pots or in field? So we started off with a millet grain inoculum to develop a kind of pot-grown Pythium violae mycelium assay. So millet grain has worked really well in getting high levels of Pythium quickly with good oospore numbers. Oospores are obviously the main spore type of Pythium and are one of the main components of instigating disease in carrots. These trials worked really nicely and we got really high levels of seedling damping off, reduction in foliage. The assay does produce stubby carrots, but we get a good reduction in root weight. So overall, we're getting that kind of yield loss reductions that you might see in a field. We get good cavity spot incidence in over 50% of carrots. We probably get slightly lower cavity spot severity than you would see in the field, with an average 2 to 3 lesions per root. But for a pot-based assay, this replicated the kind of scenario you see in the field really nicely. So we use this assay to kind of explore some resistance to cavity spot. So this is a glasshouse pot test using that millet inoculum. And we use different accessions from our carrot diversity set I described earlier. So we have kind of 2 assays you see here on this graph. On the left-hand side, we have the pot-based inoculum trial showing incidence of cavity spot. Kind of on the right-hand side, you'll see the results from the agar plug test on carrots. You can see those pictures there on the bottom of the screen. From our perspective, this is really good news. Those 2 assays correlate. Carrots with the biggest lesion size from the agar plug test also have the highest incidence of cavity spots. And you can see there on the left-hand side of that graph, we're starting to identify accessions that are showing more tolerance and resistance to cavity spot. reduced incidence of cavities per carrot. So this is really good news. We're starting to identify resistance, and we know that we have a good artificial pot-based assay. We also then went on to develop some artificial inoculations in macrocosms. So we have these concrete tubes submerged in the ground that we can inoculate with Pythium. So we use Pythium violi oospores that were produced in a sand oat substrate. And we get really good, high, consistent levels of cavity spot here, 40% cavity spot incidence with at least 6 lesions per carrot. And in this system, we don't get the stubby carrots. So we get yield loss, but not stubby carrots. You can see the top of that slide there, some really nice examples of the cavities we're getting. So when we look at doing the kind of resistance assays out in those macrocosms, you can see here that we're seeing really similar results. Really good incidence of cavity spot, about 60% severity with Nairobi there. And you'll notice on the left-hand side of that graph, CDS 25, showing good tolerance there. And that matches nicely with the pot assay on the previous graph. So we've now got 2 assays, pot-based and macrocosms, that work very well together with similar results. So we use this to then go on and start looking at some control. So metalaxyl alongside carrot varieties. First off in the pot tests. So we have Nairobi, our industry standard, Criolla, which is a very fairly resistant variety, and Eskimo, which is quite susceptible. And here we tested it. So we have our uninoculated control on the left-hand side of that graph. And then we tested the inoculum at 2 different densities, 10 milligrams per gram and 30 milligrams per gram. Essentially, what this is showing you is there are some differences in carrot accessions. So we are seeing some resistance there. But also, hopefully, you can see that compared to the untreated, metalaxyl is having a really significant effect on reducing cavities. So we then went on to look at this in a little bit more detail with additional fungicides in a macrocosm environment. Hopefully you can see there in the picture the macrocosms, the carrot foliage. So here again, we used our highly susceptible carrot, Criolla, and we had 4 treatments here. Metalaxyl, Prevacure Energy, Trisoil, which is a biocontrol, it's a trichoderma, kind of active against kind of nutrition and kind of spatial competition, and Ranman as our final treatment. So what you can kind of see here is, we had really good cavity spot incidence in these macrocosms. It's clearly a really good system for replicating the kind of symptoms that you guys are seeing in the field. So on the left-hand side of the graph, you'll see our uninoculated control. We do get background levels of cavity. It's an outside environment. Cavity spot and Pythium is everywhere, unfortunately. But you'll see there in that orange bar, the inoculated control, really high levels of cavity spot incidence. I think the most notable thing here is metalaxyl has had the most significant reduction in cavities there compared to all the other treatments. Reality is that those other treatments in blue have had very little effect on reducing cavity spot, whilst metalaxyl takes it to the same levels as the uninoculated control. So kind of in summary, that kind of brings the end of the first bit of carrots from me. Cavity spot, as we all know, is the primary soil-borne disease affecting UK carrots. In our research, we've kind of developed this artificial inoculation system, working really nicely for both pot-based experiments and these field macrocosms. We've used those assays to kind of understand that cavity spot really is reliant on metalaxyl. It works in both our systems, and with the other systems that we've tested so far, really nothing is coming close. And we've also been able to use these assays to identify some resistance. So we have accessions there showing resistance to cavity spot that hopefully we can take forward and understand the basis of that resistance. A couple of thank yous to our funders, DEFRA and AHDB, to John Clarkson and the Clarkson Group and everyone that made these experiments possible. Thank you all for listening, and I'm happy to take any questions or after Colin, if that's easier.
Yeah, I think that's what we'll do, Lauren. Thanks for that. I mean, that was a very nice summary of the science element of the disease, and that was the plan. And it's good. Microcosms is a new term to me, and it looks like it's been a real step forward in helping with the science.
Yes, they're certainly working really nicely. I think
Yeah,
many of us have struggled trying to replicate the kind of field symptoms that you see in an artificial environment. So
yeah,
to be able to replicate that severity is a really good step forward in testing for resistance and, of course, control measures.
yeah, and it's great that there is resistance being found in that work you've been doing.
Exactly.
And yeah, metalaxyl still, it's been around a while, still important. And we'll probably hear a little bit more about that from Colin. So look, thanks for that again, Lauren. And
Thank you very
what
much.
we'll do is we'll ask Colin maybe to come in straight away. Colin, if you don't mind, you might share your screen and we'll hear, I suppose, a bit from the industry and the practicalities of controlling cavity spot, just to let people know who you are again, Colin. Some of you will know Colin from his work over here, but he's an independent fresh produce consultant working with Veg Consultancy Services Limited, like I said earlier. Colin lives on a farm with his family in Norfolk and works with a wide range of crops, but specialising in carrots and parsnips in particular. He works from East Anglia up as far as Scotland, and like I said, is very familiar with Ireland, working with a number of growers on occasions over here. So Colin, thanks very much again, and we look forward to hearing your presentation.
Excellent, thanks, Andy, and thanks, Lauren. That's really good. That work's been really useful. So we're going to have a little bit of a go through from sort of more of a field perspective. We'll jump in there. I promise that's the last picture of cavity spot you're going to see from me tonight. So yeah, we all know what it looks like, hopefully. Going to go through a few of the sort of factors that increase risk, some of the control measures Lauren's alluded to, a little bit of discussion on crop monitoring, looking at some of the challenges that we face, and then we'll try and put a bit of an update, a current update as it stands with metalaxyl. It's nice pictures of tractors there because I know you all like tractors. So right, let's moving on. The risk factors, you've seen from Lauren there, it's very complex interactions. It's not one thing that tends to cause cavity spot. There's normal several parameters that have to line up. I would akin it to something like trying to play 3D chess. You think you've got one thing solved, you put your metalaxyl on or your SL, you can still get a challenge. Some of the other things you can do, I mean, these are really linked to IPM, and to be honest with you, most of the guys are doing them. So low soil pH would favour the pathogens. So the Pythium, etc., would favour the low soil pH. So we really want to be looking at that. Again, many of you will be doing it. The crop rotation, you can get down to 7, maybe 6 years on a polycrop. It's not in the ground for quite so long, but really a strawed crop, 8 years. Some guys hold it out to 10 years as a rotation. Some guys like to try and just get onto virgin soil that's never had a carrot crop, but trying to find the right soil that hasn't had that, and probably with a bit of irrigation as well, it's pretty tricky these days. So 8 years is a minimum, but you really wouldn't want to return to a field that has a history of it, particularly with a late crop. If there's a history, last time you were in there that had some cavity spot, I'd be either selecting some very strong varieties to try and put in there, making sure you've got an SL application if it's approved, which currently is not for you guys, unfortunately. So it's just something to consider. The other factors at play there, the organic voids say a healthy plant doesn't get sick. So, we just need to think a little bit about plant vigor, crop stress, particularly these weather patterns we're having these days. We have some either really hot, cold, wet, dry, all of that goes to stress your crop. Now, I know there's a lot on biostimulants and they all claim to save the world, but, might be worth having a chat and having a look at some of the amino acids if your crop is going through that. So just something to bear in mind. Another one that we'll come on to later was irrigation onto warm soils in the summer. I will come back to that in a bit more detail, but it's just something to be mindful of. And the other one, not that we grow in any of these fields ideally, poor drainage, and also your field low points, the points that always stay that little bit wetter. We'll come back to those, but again, obviously increased soil moisture is going to give you some problems. So the field selection, I mean, especially your storage crops, that's going to be your greatest risk. So you're really looking to try and have a really good 8-10 year gap on that. Make sure you take your soil test to get your pH correct. Ideally, you want to be trying to control that pH and getting it up around about 6.5 to 7 in previous crops if you can. I know that's not always easy because we're sort of slightly nomadic, and we tend to just sort of get the field in October and try and make the best of it. But it would be better if we could control, if we get a bit of lime on in a year or two in front before the carrots, because we do have a slight scab risk. We can work around it with some different types of lime, particularly the calcium carbamate, the prilled lime, we could have a look at, but we just have to be a bit careful on that scab risk if we got crops that can't be irrigated. Also, off of your soil test, we also look to sort of monitor and try and maintain and boost, if required, the calcium levels pre-drilling. Really want to try and get a bit more calcium into the soil if we can. That will also help, although I don't know it will solve it. Some people who sell calcium products might argue otherwise, but I haven't seen any data to say it solves it. But obviously calcium goes to cell strength, and if we can make cells stronger, then that's got to be a good thing. Variety selection. So, I mean, Lauren's shown you that there is some differences there. We'll come on to that, but it just needs to be bear that in mind, depending on the time of year you're trying to grow a crop for. It's not unheard of polycrops to get cavity spot, particularly in areas which have been particularly heavily carroted over the world. But yeah, it just needs a little bit of thought. I've got an if underlined there. So if you're not currently approved, I'm afraid, but the SL567A, if you can get it on, it's within the first 6 weeks of drilling, ideally at 1 true leaf with moisture. The key there is the moisture. You can get that onto moist soils or with a little bit of rain just afterwards. That will increase the efficacy of your application. Lauren said that the some soils can have enhanced metalaxyl degradation, which basically means if you used a load of metalaxyl on your spuds over in previous crops, your soil will have a better ability to be able to break it down, which therefore makes it disappear quicker and does less to protect your carrots. However, in a trial we did for Syngenta many years ago, it showed that even if we were in the highest degradation, we got down to sort of 10 to 14 days. It still had an effect on a cavity spot. So, yeah, don't rule it out just because it has been heavily spudded. As I say, that irrigation onto warm soils, July and August, I know we can't do a lot with heavy rainfall, which we seem to be getting a bit at the moment, but it's just something to bear in mind that can increase the incidence of cavity spot. And I'll tell you about a study we did on that. And the other one, which is always my bugbear, is the cavity spot didn't turn up just as the trailer reversed into the wash bay and tipped up. That came into your field a lot earlier than that, and we need to get out there once the crop's matured, and then even if the straw's gone on, and get out there and do some regular crop sampling. So we'll talk about that in a bit, but that is something to bear in mind. I mean, as a rough rule of thumb, in the right conditions, cavity spot will double in a week. So if you find it, we ideally want to get that crop gone. So we spoke on varieties. The VCS variety team have been doing some cavity spot trials, and this is the end of a 3-year study. So we've basically got a sort of a trial site which has grown carrots for the last 15 years, solidly year on year, which is obviously incredibly bad practice, but then that's why it's our trial site. And we do a replicated variety screening process of all the varieties carrot, and that is then analysed and statistically gone through to actually look at the results and mapped to make sure it's just not an area that's bad. But the data has actually given us, it's been fairly static over those 3 years. So the industry standard you can see there, I've highlighted in yellow. Yeah, you don't have to guess which one that is. It's not the worst, it's not the best. So we put that as the middle of the road. But you will see there are a number of varieties, as Lauren said, that definitely have, I'll say tolerance. I won't say resistance, because everyone tends to think that should be 100%. So tolerance. So there are some varieties which you can do now, and this trial obviously is sensitive, and you'll see that the numbers are cancelled out, or the names are cancelled out. The varieties you'd probably be looking at that tend to known in the industry to have slightly better tolerance would be your sort of Norfolk or Octavo would be two that would be very good. But there are some others, but it just kind of depends on your situation, and it needs some careful thought with your agronomist. And you can see the highly susceptible down there that had a over 6 as a score, and that, so you know, is Sweet Candle, which is the one we used as being highly susceptible, and to be fair, it was. So we spoke about crop monitoring. I got a picture of a young Alex there. That's my boy. Got to start early. You know, as soon as that crop's matured, I know it's wet and it's cold and it's muddy and you've got 110 other things to do, but taking a W formation sample across the field, looking, you know, sampling sort of 20 roots at each stage, a point of your W. If it's over 10 acres, split it, do 2, take it back to the back house, wash it, and you need to wash it. You cannot always see cavity spot just by trying to clean the roots off on a bit of foliage or a puddle in the corner of the field. So get it back to the pack house, get it washed and assess it. And then go back in 3 weeks' time, 3 to 4 weeks' time, and do it again. If you get any sign of cavity spot, then we really want to be talking to getting that field harvested if we can. It can very quickly increase, as I said. So, I mean, that gives you a bit of a rough idea. That's a bog standard wash sample QC report. One thing of interest, and Lauren did speak of it, you'll see under major defects, I've only got cavity spot to go for. So if I have got any other cavity-like lesions, the QC is going to put it down as cavity spot. And that is quite interesting, because when one of Lauren's predecessors asked for some cavity spot samples on carrots to start actually mapping the genomes and seeing what was there, I think about 40% of what the growers and the agronomists sent in as cavity spot weren't actually cavity spot. So it can be quite difficult to identify. And I don't think that's helped, because the QC says it's cavity, so the farmer says, it's all cavity. And that sometimes muddled the waters when we're trying to sort things out, but just worth bearing in mind. You know, Cylindrocarpans is another one which will look very similar to cavity, and it's quite difficult to spot it against it. So onto the research. I mean, the British Carrot Growers Association, I sit on the R&D panel, and we've been actively researching cavity spot for at least, I don't know, the past 13, 14 years. Where I told you about the warm soils we're getting irrigation, that came from a study I did. We had 30 sites, that's 30 carrot fields split from Inverness up in Scotland all the way down to Devon, organic and conventional. And we monitored them for 3 years. We had a soil moisture station in there, so looking at soil moisture, soil temperature and rainfall. And then we, at the point of harvest, we then assessed them for incidence of cavity spot. Now, that is a huge amount of data, so that's 90 fields in effect over those 3 years. And in 1 year, we had a correlation from a statistician that high levels of moisture, i.e. rainfall or irrigation, onto warm soils in July and August had an increased level of cavity in those sites. So, as I say, it's very complex, but it's just something to be mindful. Around the same time, we're not doing it anymore now, but we did try using soil PCR tests. So a bit like the old COVID test, but we had to go out to the field every fortnight, brush the soil off of the carrot roots, pack it up, send it over to Norway, and they would analyse it. I think it was a Dutch company that also said they could tell you by a similar technique pre-storage, so before you put the straw on, and predict the cavity levels in the field. Yeah, nobody does it, and it didn't seem to actually work very well. So for crops that said, oh yeah, that'll be fine, no cavity, went down with it, and others didn't. So yeah, we don't do that one. You've heard Lauren speak about the Warwick, the inoculation trials. I mean, that's been huge, because up until now we haven't been able to inoculate. Most of the field trials, nobody grows fields in carrots that's going to get cavity spot, which doesn't help the trials guys. And so it's been very difficult to actually be able to say we are going to get cavity of a sensible level in that site. So now we've got it, and that happened just pre-COVID, and then the demise of the AHDB, which is our sort of central levy gathering body for horticultural research, has really slowed the R&D. So I think the Warwick guys have really cracked it, and hopefully we'll be able to get it sorted out and get some R&D done, so we can actually start testing products, biologicals, other varieties, etc., to see if we can actually come up with a way of control. I promised you an update on SL, which is the metalaxyl. Basically your challenge was, I'm sure a few of you know, the EU didn't like one of the adjuvants. It wasn't the metalaxyl itself, it was one of the bits they mixed with it. The EU didn't like that, so they've kicked it out. Sports Syngenta on the hop a little bit, because in the UK we have a sale period until I think it's March, and then a use up period until March 2025, I think it is. So effectively we're covered for next year, but Ireland, the EU have kicked it out straight away, so you are not currently approved for application. The approval team at Syngenta did meet with the Irish PRCD, and evaluation report's been requested, which should be going over shortly. I don't think you will see an approval for next year. And even if you found a bit of paper and got an approval, having talked to Syngenta, I don't know you'd actually be able to get product. So as much as it pains me to say, I don't think you're going to have SL or metalaxyl next year, which, as you look on the back of Lauren's data, is a little bit painful. We can try and tick all the other boxes, but it's a bit scary, particularly for your storage crops. We did the work, so the VCS trials team again did an Areto study for Syngenta a few years back, and the new label was probably going to be a band spray only. I've got some clients who are currently band spraying over the last 5 years, and they have not seen an issue. So it has been as good as the overall spray. Obviously, you're only using about 30% of the product. So there's quite a financial saving in product. However, even if you've got a Bowser man and a Bowser in the field, a triple band sprayer only goes so fast. So there is an issue, particularly if you're trying to catch a bit of soil moisture. The guys tend to find that they can't do everything, but hey, we might have to get more band sprayers. We'll have to see how it goes. But when we say a band sprayer, it doesn't need to be a camera guided, shielded band sprayer. It just needs a nozzle over each drill row. An 80-degree nozzle will be fine. If you get a bit of drift, it's not going to be the end of the world. So just bear it in mind. It's probably something, sort out in a reasonable workshop when you've got nothing else to do because it's really wet outside. But yeah, so it's worked okay. But as again, that soil moisture is key. So soil moisture trumps growth stage in my book. So, you know, if it's just after drilling or, you know, one true leaf is ideal. If it's a bit early and that's cotyledon stage, but you've got good conditions and an approval, it's the time to put it on. So, yeah, it is what it is. I can't do anything else. We've pushed as hard as we can. So that was a bit of a brief tour, but I'm sure that hopefully there'll be some questions. But yeah, thank you for listening. Hopefully that's been useful.
Absolutely. Thanks for that, Colin. Great summary there of the industry side of the house, I guess. And yeah, thanks for the update on SL metal axle SL 567A, which, as you say, we're in a bit of trouble. I think it's currently approved until Christmas Eve, but we have a gap next year. But look, hopefully it'll be back to us again under the new label and formulation in 2025. But so look, like I say, thanks for that. You've given us a lot of you laid it bare there in terms of the metal axle. I say it is the only show in town, I think, really, in terms of the chemistry, from the work that Lauren's done and from your own experience. Just to say, ladies and gentlemen, again, look, there's an opportunity to pose questions to our expert speakers, and this is a great opportunity. It's your meeting, and it's a great opportunity for you to ask the experts while they're on here for the next half an hour or so. So please, if you don't, maybe any question or comment, just enter it in that question and answer tab at the bottom of your screens. We might just take one or two. There is one or two questions, I think, coming in. Owen and William, do you just want to pick up on one or two for now, and then we'll move on to the Canker?
Yep. Thanks, Andy, and thanks, Colin and Lauren. They're brilliant presentations. While we're still on the topic of metal axle, I suppose just one first for you, Lauren, if you don't mind. A question there on: are you planning on testing any other active ingredients against cavity spot and comparing to metal axle? We saw you had Ranman there, a potato fungicide, but are there any others in the pipeline?
We would very much like to. But like Colin alluded to, with the loss of AHDB, we are a little low in funds at the moment. So we're currently in the process of redoing our tender for Vegin, and we're trying to work out what we could put forward in there. We would certainly like to continue testing. We have the assays up and running now. Yes, and I'm rather hoping I can talk to Colin and the BCGA team shortly to try and see if we can get something going. But yes, if you have suggestions, we are very open and willing to suggestions. So if you think there's any fungicides that would be suitable contenders, please do let us know.
Yeah, it'd be great to get take advantage of that infrastructure, I suppose, when you have it now working well. Just another one here to Colin. I suppose, Colin, you mentioned prilled lime and the situation if a grower gets access to land around October time. Is Grand Lime or Prill Lime, is that the best way to increase pH quickly, or is it better to be in a couple of years in advance and go with ground limestone?
Yeah, it's better and cheaper to be in a couple of years in advance. And to be honest with you, I don't know why any grower, unless you're growing blueberries, would want to have an acidic soil. But I think some people may stick their heads in the soil a little bit. But before would be better. Prilled lime is very expensive, so I would say put it on not in front of destoning, but in front of bed forming, and just try and mix it in, the top area, not all the way down, but just in the top area. But you do need to keep an eye on your scab risk. If you've got a very dry field, or it's known to go dry, and the river's a long way away, you want to be a bit careful. That can increase your scab risk in dry conditions. Your other alternative is calcium sulfate. So you've got granulated calcium sulfate. So that doesn't touch your pH, but that will give you calcium and sulfur, which are both useful. It's probably a more modern version of gypsum for the guys that would be used to that. So they're the sort of products. But again, it kind of depends on your soil test and what the results are for calcium and your pH as to what product you use and the quantity you use it in.
Just while we're on that, Colin, sorry, just on calcium, is top dressing with calcium nitrate? Is that is that helpful, or like too much nitrogen probably doesn't help the situation?
Yeah, I mean, carrots are immensely good scavengers. I have grown carrot crops with less than 20 kilos of nitrogen. And we have got some guys in Ireland who grow with very low rates of nitrogen, and they still get Nairobi to come up, flop over, and go down and lodge. The calcium ammonium nitrate, CAN, or even calcium nitrate, it's very quick for getting nitrogen into the plant. It's not so sharp. I mean, before I was doing carrots, I used to grow 1,000 acres of iceberg, and we would use calcium ammonium nitrate because tip burn was a massive challenge. Yeah, it didn't make a lot of difference. So I would say you're better off with putting things underneath the plant than you are. There are a lot of people who will sell you a lot of calcium products that claim that they can spray it on and it attaches itself to another element and that element drags it through the plant. Yeah, I wouldn't necessarily agree with that.
Okay, I'll pass you over to William there. I think he might have one or two more.
There's a few questions there. Thanks, Colin and Lauren. Great presentations. Lauren and Colin, both of you. Colin mentioned about PCR test. I just wondered if there were, from a question here, if there were any further developments in molecular testing in field, or has that paused for now? It might be both of you could have a look at that.
The test I was done was Arne Hermansen in Norway, and I believe he did. John Clarkson did speak to him and have some involvement with them, but we couldn't get it to work, and we couldn't get it to work and replicate in a useful way compared to what Lauren's done now that she can. Yeah, I think at the moment that's a better place to be. But I don't know, perhaps Lauren's got some more scientific input on that one.
Very similar. So we have developed PCR diagnostics in-house. I think what we've struggled with is rather like growers, the unpredictability of cavity spot. So it's really working out the frequency of sampling has been really challenging. You can sample different parts of the field and get completely different results, or even wrong timings, and you get no positive results, and yet cavities there in a matter of weeks later. So kind of working out that sampling and timing is really challenging for the molecular work, I think. But it's certainly something that I think should continue to be explored.
Great, thanks. Just one more before finishing up here. Colin, you mentioned organic production there. Are you linking some of the differences in terms of soil health, or could it be variety differences? It'd be good just to get your opinion on that, please.
Yeah, sure. I mean, you do tend to find organics is on slightly more marginal land for roots. But having said that, I got some guys in East Anglia who've put perfect sand land into an organic situation. It just tends to be, from all the samples and yield digs I've done, you just don't tend to see. I'm not saying you don't see cavity spot in organics, but the incidence of cavity spot is so much lower. It's just an observation. I haven't got any science to back that up. Lauren may be able to add some. You'll get the stalwarts who will tell you, well, their soils are healthier, so therefore the plants don't get sick. But that's a little bit rosy tinted to me. But yeah, we do see a lesser level, lower level of infection from our end.
Yeah, I'd agree. I think it's a really interesting point and kind of highlights that we still don't really understand some of the fundamental biology of the Pythiums. It's quite clear they can work together in a complex, but we still don't really understand how they work together with the rest of the soil microbiome. So the other pests and pathogens that exist there, there's probably lots of other beneficials or pathogens interacting that are all kind of playing a role in the role of infection. Lots of questions.
we won't have time to take them all tonight, but I want to thank everyone for their contributions so far. Yeah, complexity, that's the name of the game by the sounds of it, and we'll hear about another very complex issue in Canker in a minute. But just to say again, ladies and gentlemen, if you have any questions, look, please feel free and put them in the question and answer box there, and we can field them before we finish up later on. The other thing to mention again is the IASIS points for anyone that's wanting to make sure they get credited for attending the meeting tonight. Owen has put a link in the chat box, I think, Owen, for anyone who wants to avail of the credits going with this meeting. So please fill that in before we finish up. So we'll move on, ladies and gentlemen, to our second disease, if that's all right with everyone. And again, we're going to have Lauren first up with this, followed by Colin with both with presentations, and then we'll take questions again following the presentations in the same manner that we've done with Pythium and Cavity Spot. So Lauren, if you don't mind sharing your screen again, just to say that Lauren is very comfortable with this subject, I've no doubt, because she did her PhD in it. And it's work, whilst it might have been a couple of years ago now, at this stage, Lauren is probably as up to date in terms of what work has been done on this terrible disease that we're very familiar with here, and is the bane of anybody's life in terms of those that are specialist growers in parsnips. It's a hard nut to crack. So look, we look forward to what you've got to say, and we'll follow on straight away with Colin's take on it. Thanks very much.
Yeah, thank you very much. So yes, parsnip canker, big problem, obviously. Today, I thought I'd just kind of go over kind of understanding the causal agents, so the pathogens responsible, and kind of talk a little bit about breeding for resistance. As Andy said, parsnips are one of my favourite subjects, so please do stop me if I chat too long. I could chat for hours and days about parsnips. So parsnip canker, as we all know, is one of the biggest problems for parsnip growers in the UK. Literature estimates losses at 1 million per annum. I suspect it's significantly higher than that. Growers will regularly comment on 80% crop losses, and it's approximately £64 million annual economy. So it could be significantly higher than that. Parsnip canker was kind of originally attributed to the fungus Itacinelia back in the 1950s, in the kind of post-war era. I think now it's kind of accepted that canker is a kind of umbrella term for a range of symptoms and pathogens. I think one of the key things we do know is that it is exacerbated by insect damage, particularly carrot fly. So controlling insect damage is key. So again, what are the problems? Well, first of all, is identifying the problems, the causal agents. We know that there's more than one pathogen playing a role there, but kind of the relative severity of each of those pathogens needed to be understood. And then once we've understood those key pathogens, it's looking at those populations. These are very much what we would consider, I guess, niche or novel pathogens. So there's not been extensive research. So we need to understand that those populations are kind of static. We don't have any regional variation. And to start to understand a little bit about the biology of those pathogens to give us an indication of time of infection, etc. And then, of course, kind of develop some assays so that we can start screening for resistance, start looking at control methods, and then from a kind of breeding company perspective, providing something that can be integrated into a marker-assisted breeding strategy. So starting off with the causal agents of parsnip canker, I got sent diseased parsnips from predominantly around the UK, but actually around the world, including samples from France, Germany, Spain, New Zealand, Norway. And hopefully you can see from this graph here that the kind of predominant pathogens that we were finding really were cylindrocarpin and Mycocentrospora, with Itacinelia to a much lesser extent. You'll note there in green, Fusarium does come up quite high. I'll, in the next slide, explain that a little bit further. Other pathogens to a much lesser extent, about 10%. And these include things like your Pythium, again, with the cavity spot, botrytis, foma, your kind of general soil-borne pathogens, but none of them causing such major problems as really cylindrocarpon and Mycocentrospora. As you saw there, Fusarium, about kind of 27%. Is it a big problem? Yes and no. Fusarium is an interesting one. There are many, many species. And as you can see here, I think there's over 15 Fusarium species were identified from parsnip canker. I think it's worth saying that Fusarium very much tends to be a secondary pathogen. It doesn't, or it very rarely seems to have the ability to kind of do the initial breaking of the skin and cause the initial symptoms. It very much tends to come in as a secondary. We know Fusarium is prevalent in many crops as a pathogen, so it's really not the primary cause here. We really are, I think, focusing on that cylindrocarpon, Mycocentrospora, and to a much lesser extent now, Itacinelia. So I thought I'd go into those three pathogens in a little bit more detail. Itacinelia, first of all, so we consider a basidiomycete fungus, and it's technically comprised of 4 species. This is only really important because historically, according to the literature, only Itacinelia pastinaceae, so only isolates of parsnip, should be able to infect parsnip, and isolates of other hosts like sunflower or the herbs shouldn't historically be able to infect parsnips. It is a seed-borne pathogen, which is why it's really important all the seed companies are now washing their seed and pelleting. That pretty much solves the problem, but it is important to bear that in mind. The pathogen produces ballistospores and chlamydospores. So ballistospores are those ones that will cycle around the seed and the foliage and kind of cause those necrotic lesions in the foliage to fall down on the exposed crowns of the parsnip roots. Chlamydospores are very much the long-lived spore types. They can survive for a long time in cold conditions. Itacinelia is actually quite closely related to Arctic yeasts, so really can survive in very cold conditions. And from my own research, I know it will happily grow at 0 degrees. So winter is not a problem for this pathogen. As we know, it produces both foliar and root symptoms. So that stereotypical kind of brown necrotic lesion on leaves surrounded by that green yellow halo. And on roots, the cankers tend to be black, very dark brown, and quite a hard canker. You don't really get the soft, mushy cankers with Itacinelia. Cylindrocarpon is an ascomycete fungus. It is relatively closely related to Fusariums, and it causes those very characteristic orange corky cankers. It's a soil-borne pathogen, and again produces those resting chlamydospores that are again very tolerant of cold conditions for a long time. Again, why it's really important that we have extended rotation periods. It's got quite a small host range. Grapevine and gingseng not going to be a problem for us. Pine trees can be. Anecdotally, some Scottish growers have had problems where they've got fields next to extensive pine or spruce forests. And I've had people mention that they occasionally use wood chippings as soil stabilizers, which we think could be potentially bringing in cylindrocarpon to fields. So kind of just something to be aware of. Mycocentrospora is the third one here. Again, an ascomycete fungus, causes dark sunken lesions. These are kind of the more black, slightly purpley, can look around the edges. More of a squishy canker this is. So Itacinelia tends to be hard, Mycocentrospora tends to be squishy. You of course get it in carrot as well. So again, this is why we need good rotations. It's a soil-borne pathogen, again, very happy in the soil for a long time at cold temperatures. It has a slightly wider host range, so you will find it on other commercial crops, carrot, lettuce, herbs, and it will happily survive in weeds of Asteraceae and Brassica, as well as the Apiaceae. So kind of edgeway hygiene is really important as well. So in terms of results, I thought I'd start by talking a little bit about the characterization of these pathogens. It's actually really important that we understand the pathogen kind of populations, because when we're breeding for resistance, we need to know if we're breeding for resistance to a single race and a single population, or if perhaps we're seeing different populations across the country. So we started off by developing a root assay. This is where we inoculated parsnip roots with an agar plug of the fungus, in this case Itacinelia. You can see in the middle there forming lovely dark cankers on the crowns of the roots. And on the top right-hand graph there, you can see lesion area. Very little difference there between the different, so 50 odd different Itacinelia isolates we tested there, and very little difference. One thing to note is the slightly darker bars are the isolates of non-parsnip hosts, so things like carrot, herbs, chrysanthemum, a couple of other flowers. You can see that they're equally dispersed amongst the parsnip hosts there. Next up, we developed a leaf virulence assay. So it's important to look at the leaves because, of course, Itacinelia produces those two spore types, and the ballista spores are the type that are circulating around the foliage and seed. So we need to understand if we're seeing kind of different levels of pathogenicity between the root infection mode and the leaf infection mode. So here, leaves were inoculated with a spore suspension, and you can see the results here. Slightly more variation, but again, the key thing here is that we're seeing is those non-parsnip hosts are mixed in evenly amongst the parsnip hosts. So in terms of that historic literature where it says that only parsnip isolates should infect parsnips, that's perhaps not very true, and actually Itacinelia off any crop could be a problem for you. I know it's not helpful for you, but in terms of breeding for resistance, this was important for us to know. So next up, we've got cylindrocarpin. Again, testing the virulence of the pathogens here. So we've got 16 cylindrocarpin isolates on roots. Again, that top right-hand graph, you see a small range in lesion size. There's only 1 centimetre square difference there, which is quite small really compared to some of the cankers you'll see out in the field. And next up, we developed a seedling virulence assay. So cylindrocarpin doesn't cause foliar symptoms, but it does cause this kind of brown root symptom in seedlings. Hopefully you can see the little blue box around the base of that seedling there. It causes essentially a canker symptom, but on a seedling. So by inoculating sand and compost with cylindrocarpin, we can induce this brown root symptom. And that's kind of the kind of damping off symptoms that you might see in germinating seedlings in your field. And you can see there we get slightly more variation in the isolate behaviour. But essentially what this is telling us is that we do only have one population of cylindrocarpin to worry about. So if we use a representative isolate in control assays or resistance assays, we're going to get accurate results. And finally, looking at Microcentrospora here, again, the top graph is our root inoculation assay. See lovely big cankers developing there. From our perspective, we do like to see a good canker in an experiment. Very little difference. And again, in the seedling assay, using an inoculated soil compost system, there is like no difference between the isolates. So from our perspective, this is really good news. It means that kind of whatever isolate we use for future assays, we know that we're going to be getting a representative idea of what's happening across the country. So we can now use these assays to start looking at resistance. So here you can see we've got 10 parsnip lines. And this is again that seedling assay with Itacinelia and Microcentrospora. On the left-hand side, control is in the grey and our inoculated is in the blue. This is obviously because you do get some damping off and seedling death, even in your control. And then, of course, on the right-hand side, we have Microcentrospora in the red. What's really interesting to note here is that our kind of most susceptible parsnip in Itacinelia P15 is actually our most resistant in Microcentrospora. So this starts to tell us that the mode and mechanism of resistance for these different pathogens is different. So when we're looking to identify potential tolerance or resistance, we're unlikely to find a single parsnip line that will be resistant to all of these pathogens. So we followed this up then with the root resistance assay, again using those agar plugs onto the crowns. And you can see, hopefully those pictures there, this is of the Mycocentrospora, those pictures. On the left-hand side, we have our susceptible root, and on the right-hand side, you can see the more tolerant root. And the difference in lesion size is really obvious there. So from our perspective, this assay is working really, really nicely. What's interesting to note is in terms of the kind of the root tolerance, we see far less significant differences between the modes of action. So you can see there starred is P15 in the Itisnilia, just below the midway point, and P15 in Mycocentrospora is only a couple down. So potentially slightly different modes of resistance when you're thinking of damping off in seedlings compared to parsnip roots. But developing these assays are really the key to kind of screening for resistance. And we then use these to move on to a much larger kind of mapping population, a breeding population of Elsams. We screened a lot of parsnip roots, about 12,000 parsnips a year. So a lot of inoculating and a lot of photographing and a lot of measuring lesions. But we got some really, really good data. And hopefully you can see from this graph, we get a really good range of resistance and susceptibility. You know, some of those parsnips, they're showing really good tolerance with kind of only 1 centimetre per square lesion. And I know that still sounds big, but you have to remember that we are really pushing the kind of disease pressure here. We are forcing a canker to develop. And by having this kind of range of resistance and susceptibility, it gives us real options for crossing and starting to identify that mode of tolerance and mode of resistance. So these kind of assays really provide the breeding companies with good breeding material, essentially, to take forward for breeding, marker-assisted development, and to hopefully produce some tolerant varieties for you guys. So it was very much a whistle-stop tour of parsnip research, but hopefully you can see that we've really identified Mycocentrospora and Cylindrocarpon to be the primary causes of parsnip canker in the UK now. Itisnilia is still present, but definitely not as prevalent as it once was. And that's probably in part, well, mostly in part, due to the seed companies and their treatment of the seed now. So simply washing the seed will knock off pretty much all of the Itisnilia that might be on the seed coat, and certainly pelleting will remove it all if there's anything left behind, which, to be honest, there very, very rarely is. We've developed seedling and root inoculation assays to monitor pathogen populations, and we've determined that there really is no race structure in these. So focusing on a single isolate of these pathogens means we'll be understanding resistance. We've got good resistance screens in place, and we've managed to identify resistant parsnip accessions. And that's now moving forward with the breeding companies to integrate those into those commercial lines and start to understand the mode of resistance. Huge thank you to Elsams and BBSRC for funding, John, Guy, and all the Elsams team for all their help and support. Thank you all very much for listening. I'm happy to take questions after Colin's.
Yeah, great teamwork there by the sounds of it, Lauren. And it's unusual to get someone that gets excited about parsnips, because we're generally are a canker on parsnips because we get the other end. People go into fits of major depression as we do when we see crops infected with this awful disease. And
Yes.
but I can see the importance of the assay work. It'll hopefully lead to better varieties going forward. So yeah, look, with that, we'll go straight to you, Colin, if you don't mind, and we'll hear what your take on all this is from the grower's perspective, I guess, and an agronomist perspective and in the field. And then what we'll do is we'll hopefully we'll get some time for a few questions at the end. So thanks very much again, Colin.
Yeah, okay, cheers. Thanks, Andy. Yeah, so Lauren, you really do get excited by parsnip canker. It's worse than I am, but yeah, we'll give it a best. So what we'll go through similar sort of format, the factors, increasing risk, see if we can reduce some of those through, IPM control measures that we've got. We'll look at some of the variety trials that we do and also some of the lists. So we'll move on. The traditional one, as Lauren alluded to, Itacinelia. Got a number of chemical options on the foliar phase for this one, so which is quite useful. So that's Amistar Top, Signum, Switch, and the Tevo and the Rudis as the trade names for those. We'll have a little look at application rates and stuff and timing, but ideally that's what we want to look at. And cylindrocarpon, which you've heard quite a bit about. Unfortunately, we don't currently have an agrochemical control or a biological one that we know of. And under the the review of organic matter amendments, that does relate to people putting wood chip on. And as Lauren said, can prove challenging, is the thought. So just something to bear in mind. And also, again, look at your pH. Keep your pHs up. We're in a sensible area. So some of the risk factors, something that sometimes quite often scares me is you look at last year's parsnip stubble, which has sort of ended up back to the landowner, and you can see parsnips flowering through the season, and he hasn't bothered to kill them off. We really do want to sort of bit of housekeeping there. Don't really want to let those spores from that crop then move into our crops nearby. So let's keep on top of those if we can, guys, and encouraging to get them well and truly incorporated and sprayed off. As Lauren said, the varieties, susceptibility, it's a tricky one because we're not talking about one infection. I think Lauren even showed you a slide that said one variety was good on one and not so good on another. And potentially we're also looking at Itacinelia and Pythium as well, possibly, all sort of getting lumped down as a canker. So it's a little bit difficult to work through that, but there is some options. Again, Lauren said poor seed quality, so Bijou, or it's Elsams who supply the Bijou parsnips, and Tosas, the Javelins and the like, they're pretty hot on it now, and actually Elsams do all the seed work for Tosas anyway. So I was actually at Elsams on Friday looking around the seed plant, and they're very strong now on washing seed, testing for any pathogen and cleaning. So that is very useful. Again, we've mentioned previously, reduce plant stress and vigor. Let's try and keep these crops going forward if we can, but not necessarily pouring large chunks of nitrogen on them. And also maybe a potash. We could look to split the rates and do a slightly little and often rather than great big lumps of nutrient going on. Just let steady growth rather than trying to have rapid growth. Again, Lauren mentioned other damage to the root, which enables the infection to get a hold. Scab and carrot fly too. There, we're pretty good. Well, we should be pretty good at looking after carrot fly now. We've got enough options, so that should hopefully be okay. Inadequate fungicide program. It always scares me when someone says, "Oh, I'm going to do reduced rates and I'm going to try and save a few quid and cut back on my fungicide program." I've never known it to end well, so I wouldn't be an advocate of that. And we now have such a strong level of fungicides. I think that's helped to reduce with the seed washing the incidence of Itacinelia that we've got. But there are some things we can do there. And again, the application rates, we want to be look at the target you're trying to hit at the time. If it's a small plant, you want to be thinking about angled nozzles, forward and backwards to get good coverage. 200 litres is normally fine. But as that canopy gets stronger and denser, probably looking, up to sort of 400 litre rate. want to get good coverage and potentially even the veg nozzle then to get better penetration into those lower leaves. So there's a bit we can do there to make sure that the money we're spending is actually being getting to where it needs to be. And again, waterlogging, it's low spots. It's probably why in recent years we're seeing a lot of it. We have some pretty high rainfalls over in Ireland, and sometimes when you can't even get the driver to the harvester in the field because it's too wet, you're going to start seeing canker after that of some type. It's just that the environment is just so favourable for infection that it doesn't really matter what we do at that stage, it's going to hurt. So let's have a look. Field selection, especially your late crops again. We want to have a good rotation, keep that well away. I'd still probably hang around 7 or 8 years. Get on top of that parsnip trash and desiccate it off, get it sprayed off. And the other one sometimes can cause us a problem is when we start dumping packhouse waste in fields, because obviously that's all the cankers or the rejected ones then get tipped up in piles around the field. Or I have seen issues where cattle have been fed packhouse waste spread around the field, and where the tractor tipped up, you can then find that in following crops of carrot or parsnip. So just something to bear in mind. The tolerant varieties, obviously Lauren's works at Elsams, and those guys have really led the field. Tozers as well, trying to get some new varieties out, which we are starting to see now with the likes of Teagasc and Phantom. But as I understand, talking to Elsams, the tolerant varieties are tolerance they're trying to look at for cylindrocarpins and microcentrospora. So we need to be looking at other measures as well to keep the whole range of canker-causing pathogens at bay. And one of the traditional ways is when we're going through with the hoe is actually to try and get a duck foot tine in there if we can, just to earth the parsnips up. And we might need to do that 2 or 3 times. I'm not talking sort of ridging them up so you can't see any leaves. I'm talking about putting an inch of soil on top, up around the crown. Well, the crown's fairly socketed in. But that's one of the things that they'll look at, is trying to find varieties now that have much shallower crowns, so you don't have that receptacle to literally catch spores of Icynilla washing down from the leaf, and B, just to sit there and bruise nicely as it going through the system. So you'll tend to see shallower crowns are coming. Again, crop stress. So let's try and keep the crops going nicely. One of the studies we did look at, as Syngenta did a study a few years ago now, where they actually extended the fungicide program. So normally you would expect mid-September to the end of September would probably be a standard cut-off period for fungicide application. But it was very evident that if you put an extra application on, into October, the first week of October, you kept the canopy cleaner, and they saw a reduction in that site of canker incidence. So I'm not saying it's perfect, but it helps. And again, if you have got, if we are looking at Pythium infections, the only Pythium approved product, well, it's not approved for you at the moment, but is the SL, is the only one that currently will help with that. So we host, the VCS trial team host a biannual event. You've probably heard of the British Carrot Growers do the carrot variety event. Well, this is the parsnip side of it. It's not quite such a big club, but we do what we can. You can see the picture in the background is this year's. We were supposed to have a few chaps coming over from Ireland, but I think they went down with COVID, so I didn't make it this year, but definitely come over in the past. And we grow in a commercial field. This was at Brandon, just outside Thetford. And we have a replicated plot trial so that the breeding companies, i.e. Elsham's Tozer, a little bit of ProVeg, give us the varieties they want us to sow, and we will then do a replicated trial. So they're all drilled the same drill, same depth, and it enables you to actually see them growing in a commercial situation. So, as I said, the newer hybrids from Elsham's and Tozers, we're looking at increased Microcentrosporans Lindocarpon tolerance. I will say tolerance and not resistance, because people tend to get a bit upset when they grow a resistant or a tolerant variety and it doesn't have 100% resistance. I've got Pegasus there, and Tozers have got a new one coming out, which is still coded at present, TZ5152. Which seems to have pretty good canker resistance again, but that will be for Microcentrosporans Lindocarpon. We've spoken about the shallow crown, tends to be better. And the other interesting fact I didn't learn until the other day was traditionally the whiter root parsnips, which is the white spear and the likes, which everybody loves, including the supermarkets. They're all so lovely and white, actually have thinner skins and therefore a greater risk of cankers, whereas your yellower roots tend to be slightly thicker and slightly more tolerant to canker infection. I've got seed treatments down there. We're a bit limited. Obviously, we've lost metalaxyl as a seed treatment. The UK are going through as an Article 53 emergency approval to see if we can get seeds treated for this year. But we're in the hands of the gods and CRD as to whether it turns up on time, if at all. Seed washing, Lauren's spoken about, so that's fairly extensive. And your only other option then really is fluidoxonil, which is Maxim. Maxim won't have any effect on Pythium infections, so it's limited. In some crops, spinach, there has been shown to be some phytotoxic effects. I haven't overly seen that in carrots or parsnips, but I would always, particularly on parsnips, urge you to prime your seed, use prime seed and use it fresh. Don't dig a sack out the office that you left over from last year. It won't be so good. So yeah, use fresh prime seed, and ideally the largest seed that you can get hold of and drill. Have a bit more vigor, and if we can't get these crops up and through the ground. Right, this is the list, just to give you an idea from this year's, it's not a complete one, but the sort of things that we were looking at, excuse me, from the variety days. This goes out to the clients. And we're looking at definitely a lot more shallower crowns. So that takes us to the end. Have we got any questions?
Thanks, Colin. Absolutely. Listen, as ever, some great practical advisory tips there for anyone in the business of growing parsnips and trying to deal with this disease. It's one of those ones, I think it's so complex, and it is an integrated approach, like you're suggesting, a mixture of actions. I think there's one or two questions. We're getting close to closing time, but we'll take one or two. So I'm going to go straight over to the lads. Owen or William, you might have a couple of questions for
Certainly,
our
Andy.
speakers.
Thank you very much, everyone. Great presentations again. We've got a good question in here from the audience. Do you think there's a correlation between lighter soil types and higher canker infection?
Lauren?
Possibly. So we think possibly soils with greater drainage might have fewer spores kind of staying in there for longer. That might help reduce things. Again, there will be a whole community of pathogens going on. So a kind of less organic material might have a slightly lower kind of pathogen population and microbiome going on, which will affect canker incidence. Colin, you've probably got a better idea than I do, though.
Well, I mean, all I would say is that predominantly most of the parsnip land in Ireland is probably heavier than the parsnip land in East Anglia. I would see more canker in Ireland than I do in East Anglia. But I also think you have higher rainfall levels and soil moisture levels than I do in East Anglia. So we've seen, varying soil types. But as I said, as Lauren said, it's such a complex thing. If it were everything equal, you may well find, yes, it does. But with the, soil moisture levels being that much higher, that's not in favour of the crop. So I don't think if you, out of the two fields in Ireland that you choose, you could almost argue, if I got a sandy field, will it drain quicker? But then is it going to get stressed in the summer if it's a long way from the river? There's so many factors that could affect it throughout the parsnip's life, if that makes sense. So I think good husbandry, good housekeeping, do the basics, and that's pretty much all we can do at this stage.
Thank you very much. I've got one more and I'll hand over to Owen. Does higher planting density or later sowing dates produce canker levels?
All I would say from a practical point of view is if you're trying to store a crop or take a crop longer, it tends to be more mature crops that suffer infection. A more immature crop going into a storage period I wouldn't say it's perfect law, tends to be a little bit more resilient to some of the sort of rots and the cankers and the breakdowns. So I would say, more immature crops better, but you obviously need to have yield there, otherwise it's not much point. I haven't seen density greatly affecting it. I've seen plenty of canker this year on crops which are very poorly established with damping off. So that's from my angle. I don't know if Lauren's got anything to add.
No, I don't think we've seen any kind of impact on density at all. As we know that pretty much all of the key pathogens, Cylindrocarpum, Mycocentrospora, and Itis, they're all very comfortable surviving at cold temperatures. So, getting it in later, the pathogen's not going to have a problem with that at all. It's going to be very happy to infect and munch away. So perhaps not always the best plan from the pathogen kind of biology perspective.
Great, thanks guys.
I just have a couple coming in here as well. Thanks very much, Lauren and Colin, for great presentations again there. Colin, you mentioned covering the tops of the parsnips and possibly going through maybe 2 or 3 times. That's kind of the number of times you'd kind of have to typically go through. When would you recommend starting that usually?
So I'd probably be looking to start that around about the sort of 4 to 5 true leaves. When you start thinking about putting a fungicide on the crop. I mean, it's a traditional control for Itis, but if we haven't got an Itis problem because we washed it all off the seed and there's nothing coming in from any neighbouring fields, and you've got a good fungicide program, it's a bit academic, but it's something positive, proactive that we can do. And if we're going through, which we will be, into row because we haven't got a lot of herbicide options, it's just one to think of, for the sake of running a goosefoot tine through there, just to put a bit of soil around them with you kind of regret not doing it if it's a problem later on.
Yeah, we have a question on fungicide programs as well, but just first on while we're still on weeds and into row, I suppose maybe this one's probably for you, Lauren and Colin. I suppose in relation to weed control in the crop and the inter-row cultivation, how much of an impact does weeds in the crop have on canker?
It's an interesting one. It certainly will play a role. So any APAC weeds will, Itis will happily grow and proliferate on them. And that's certainly where you need to be careful with foliar symptoms and that necrotic tissue falling down on exposed crowns, which again is where kind of hoeing up comes into play. Mycocentrospora has a very wide host range, including many of the common weeds that we would see, any of the kind of Asteraceae and Brassicaceae weeds. It can probably have a go at, and it can persist in very low levels in them. So you won't necessarily see any symptoms but it will just be persisting in the roots. So weed control is probably very sensible at trying to reduce. We're really trying to reduce those long-lived spore structures in the soil, because once they get in, they'll stay there for a long time, and it's really hard to get rid of them. You know, the fungicides often can't reach those parts if they're really deep and long-lived. So good weed control will hopefully kind of manage that. I don't know if Colin's got any thoughts.
No, I completely agree. I've never known anybody to overly want any weeds in their crop. It's quite an interesting point, actually, because I went out with someone from the sort of approval industry this year, and the point was made, well, you've just got to learn to have weedier crops. They don't need to be, they don't all need to be spotless. And I did raise the point that two things for integrated pest management. Yeah, that's great for this year. When we come back round again, the crops that we've left behind have now seeded and gone into the weed bank. So that problem's getting worse. And as Lauren's just said, potentially you're actually leaving a host for pathogens that's going to attack your crop later on in the soil. So, it's tricky in parsnips because we are very limited to weed control options, very limited. But yeah, I would totally agree. I've never known a weedy crop to be useful for anything.
Thanks very much. And just one final one, because I know we're running over now, but it's good to answer these questions while they're there. Colin, you mentioned starting the fungicide program at 4 to 5 true leaves there, I think. And just very briefly, are you starting your fungicide program along with your insecticide program? What is the ideal timings and how long are you spraying for? What's the period that you're starting in a nutshell?
Yeah, no, for sure. Well, the insecticide program is pretty much looking at carrot fly, maybe some cutworm issues, depending if you've got irrigation or not. So that timing is weather dependent, and we run the Rosemary's model for these sort of first and second generation. Now we've lost false treatment, so that's fixed. That's one date that's carved in stone. It's really the early open ground parsnips that I would start to treat with a fungicide, probably in front of the carrot fly or second generation carrot fly treatment. It really depends on the foliage. It's as I'm walking the field, if I've got starting to get a reasonably dense foliage, obviously we're talking about Slingercarpens, Microcentrosporitis Nilia today, but you've got powdery mildew and various other issues to think of. So it can be as simple as starting off with a good application of sulfur for some of those. So it kind of depends on what the weather's doing, what the crop stage is doing, and then we splice it together once we know when the carrot fly are coming in. So you're only running through once. But I did mention it earlier to Lauren, I think, but we haven't got any disease prediction models for these infections at the moment. So we've only got once a fortnight because we're going through with carrot sprays. In theory, in other diseases, you would say reduce the interval during high period of risk, but I don't know if we've got that detail yet. So yeah, it tends to be a fortnightly spray, but there's lots of good fungicides out there now. So a well-timed spray can work your way through and then up to the first week of October.
That's brilliant. Thanks, Colin. Thanks, Lauren.
Thanks very much. I think it's closing time, ladies and gentlemen. If we were in the pub on a Friday night and someone shouted if there was time for one more, I'd have to say not on this occasion, right? But look, really very interesting evening from my point of view, and I want to, on behalf of our audience and behalf of ourselves in our Teagasc Vegetable group, can I say a big thanks to both Lauren and Colin. And just to say again, we have recorded this and it'll be put up in the YouTube channel, I think probably within the next week or so. If you need to go back and watch it or let people know that mightn't have been able to join, they can watch it there. And that'll be accompanied by the slide presentations. Both Lauren and Colin have been generous enough to offer the presentation, so they'll accompany the YouTube clip. So thanks very much for that, guys. Really appreciate that. One final thing before we finish, I think, Owen, you just want to mention another webinar that we're having later or tomorrow, isn't it?
One tomorrow, yeah. Thanks, Andy. We've another webinar on in downy mildew and fusarium in protected and field lettuce for those that are interested. Tomorrow at 7 o'clock. You can register online. I think most people that would have been invited to this webinar would have received a link, but for those that are interested, that's happening tomorrow at 7.
Great stuff. Okay, and with that, listen, thanks again, and all I can say is good night and safe journey home. Thanks very much. God bless.